Hello my students and welcome back to Excellence Batch, and I am your Diksha ma'am. So we have studied immunity, the adaptive immunity in the previous class. Now it's time to move further and talk more about the adaptive immunity. In the previous class, we have also discussed about antigen and antibody. So a lot of things will be related with them. So I will advise all of you to please first go back and watch the lectures of antigen and antibody, and then we'll get started with this one, right? So let's get started with the adaptive immunity and let's discuss more about it. So the first topic that we are going to discuss is type of immunity on the basis of source of antigen. So in the previous class, what we have done, what we have covered, let's take a look on that first. In the previous class, we have done what is adaptive immunity. Okay, what are its features, what are the cells that are responsible for it. We have also discussed what is an antigen and what is the antibody. So as you know, antigen is something glycoprotein or lipid that is usually present on the surface of pathogens or any cell, and that can trigger the immune response, and as a result immune response will produce the antibodies, right? Now we are going to talk about the types of immunity on the basis of the source of antigen, that means from where that antigen is coming. Okay, so we have two types of immunity. This is very important. Every year they ask question from this topic. We have two types of immunity. One is a passive immunity and second is the active immunity. So what happens in the active immunity? Your body meet the pathogen, or the pathogen enters into your body and your immune system come across the pathogen and produce the antibodies of its own. For example, I met with the chickenpox virus. The chickenpox virus entered into my body. My immune system recognized it and my immune system started producing antibodies against it. So this is how the source of antigen was, like it was from pathogen that has entered into my body and my body produced its own antibodies, my own antibodies, right, by working of my own machinery. Second thing, what if the pathogen does not enter into my body, but I take ready-made antibodies from other source? Imagine I am immune to some virus and other person is not immune to some virus. Now from the same virus other person is not immune to. Now the antibodies from my body is taken out and it is given to the second person. For second person, that will be the passive immunity, but my body produces the antibodies by exposure of the pathogen. So that is the active immunity. In short, I would like to say in active immunity, the antibodies or immune cells are produced on exposure to antigen, or pathogen. Now this is of two type. One is natural, another is artificial. Now for example, if I come across a disease or I come across a chickenpox virus during a disease, my body will produce antibody. So naturally I come across a disease, I didn't plan for it, right? I didn't deliberately added the virus. So that was a natural active immunity. So whenever, for example, a person got COVID, right? Naturally person got COVID from other patient or any other infected person. So the body will started producing its own antibody. That's the active immunity. Fine. Now what about the artificial? In the artificial, you are not giving the disease pathogen, you are not giving a disease, rather you are giving just the part of the pathogen so that the body start producing antibody. For example, if I didn't get the COVID, right, but I took vaccine. So what is present in the vaccine? In vaccine, we have that dead virus. Okay, and the body is exposed to the virus, but virus is not causing you disease, unlike that of this natural one. So in the natural, you got the disease. So during disease, you get exposed to the pathogen. exposure of pathogen. So that's a kind of a natural active immunity. We are making antibodies from our body, but by exposure naturally from that pathogen. In artificial, the answer is vaccine. You are giving just a part of the pathogen so that the response that is produced in the natural will be as same as here also. Okay. Then we have the passive. In passive immunity, the ready-made antibodies are taken. The ready-made antibodies are taken from external source. From external source. How? This is also of two types. One is natural, another is artificial. You know the example of natural. Like the baby can get IgG antibodies from the placenta from mother, through placenta from the mother. Also, the baby can take IgA antibodies through milk from the mother. So, the mother's ready-made antibodies are going inside the fetus. Fetus is not producing its own antibodies. So, this one IgG through placenta and IgA through milk, and that is from mother to fetus. So, that is an example of natural, that is working naturally. You are not doing anything, it will come. Okay. So, that is a kind of a natural passive immunity. Whereas in the artificial one, like we have snake anti-venom. If you have heard a person is bitten by a snake, they are sent to the doctor and doctor give them injection. So, what is in the injection? So, we make these antibodies against a particular venom. You know, understand what is venom? That zehar of the snake. So, they will always ask, if for example, you get bitten by the snake and they will ask, okay, tell us where do you live? In which area do you live? What kind of a snake it was? What was the color? What was the scale? So that they can identify which snake it was. Okay. Why they are asking area? Because they know in this area particular type of a snake are seen. Okay. Now, on the basis of that snake, they have the antibodies produced against the venom of that snake. Right. For example, if the snake is, say it be X. So, X is producing X venom. So, we will be having anti-X antibodies against that venom. Okay. So, they are given and they are the example of artificial because we are making it artificially and they are produced in the organism known as horse. Right. So, in the horse equus, right, it is produced there. Okay. Another example is antibodies during bacterial or viral infection, during bacterial or viral infection. For example, if someone is very old enough and not able to recover from COVID, the person who has recovered from the COVID, their antibodies are given to the patient who is very old and is not able to get it. So, like you must have heard of this, they are donating plasma, right. So, why they are donating it? Because that may contain the antibody. Another thing, like when someone got or met with some accident or get cut, they give the anti-tetanus serum. So, anti-tetanus serum is also the antibody against the tetanus, okay, Clostridium tetani. So, these are the two types of immunity and they are very important, and the classification is on the basis of from where are you getting the antigen. Are you getting naturally pathogen inside body during disease, like a person got COVID and now the antibody is produced, that is natural active. A person is taking vaccine of COVID, that is again active because you are producing antibodies, your body is doing work, your body is producing antibodies for your body, that is but artificial because you are taking it artificially. In the passive one, your body is not producing anything, the body is not producing immune cell, body is not producing antibody, rather you are taking ready-made antibodies from the other source, naturally like mother to fetus, artificial like that. It is just like active is just like you buy vegetables, you cook food at your home, you burn the that stove, you cooking everything and then you are eating it, right. So, you are cooking everything at house and then you are eating it. And second is you are doing Zomato or Swiggy and you are taking ready-made food and you are eating it, but you are not cooking at home, fine, just like that. So, these are two types of immunity. Let us talk about two types of immune response. So there are two types of immune response, guys. One is primary or amnestic, another is secondary or anamnestic. Okay, now what's the difference? Listen to me very carefully. This is also important. From here the questions are asked every year. Primary or amnestic response. So this is the immune response that occurs when you are exposed to the pathogen for the very first time. For example, you are exposed to the chickenpox virus for the very first time in your life. You don't have any vaccine of chickenpox virus also. You are very little, you get exposed with the chickenpox virus. What will happen? The B cell will identify it, produce plasma cell, produce memory cell, but in meanwhile doing all this thing, the virus is powerful and it will cause you a disease. So in the first exposure of the pathogen, your immune system was not that working good. The immune response was not hyped or intensified, it was very low because B cell has never seen the pathogen. It was a first time for the B cells or immune system that how the pathogen it looks like. So the immune system is not ready and the immune response is less intensified and a person usually get disease. That's a primary immune response. Okay, you don't understand? Let me explain you. For example, this is you. You are very happy, you are playing cricket, but one of your friend had chickenpox, you get exposed with the virus and the virus entered inside your body. Now, the B cell will now identify the virus first and B cell will produce two type of cell, plasma cell and the memory cell. This is B cell. Okay, but in meanwhile you will get a disease. At this time the immunity was not working that good because it does not know how the virus looks like. So that is a primary immune response. Okay, now let's talk about secondary one. Now you already have antibodies and memory cells. You already have antibodies and memory cells because you have already been exposed with the virus earlier. Now imagine the virus entered inside your body. Now these antibodies are already formed. They are already present in your blood. The virus is when it enters inside your body for the second time, the antibodies which are already which have been preformed during first exposure, they will bind with the virus and it will kill it. Now tell me, where is your immune system more efficient? During second exposure, right? And that type of immune response is known as secondary immune response. So primary immune response is the immune response that occurs during first exposure. During first exposure of pathogen, and here it is less intensified. That means it's not working that much good, you will get the disease, less intensified. But the good thing is memory cells will be formed and antibodies will be formed. Right. Now what about the secondary or anamnestic? Here it occurs during subsequent infection. Subsequent means second, third or fourth infection. Here it is more intensified immune response. More intensified is not for the virus, it's for our body. Our body's immune system is working intensively, right? It's working in a better way. Here antibodies are already present and memory cells are already present. And kill the pathogen. And kill the pathogen. So this is how your body reacts towards the second exposure, and that is usually less intensified. And this is why it is said that if you get chickenpox once in life, you will not get it for the second time. Why? Because we have already produced memory cell and antibodies against the virus. If it comes to the second time, then it will not be able to cause you disease. But ma'am, why do we get flu every year? Okay, why some people get COVID for the second or third time? If in this COVID wave you have seen, the COVID virus get changed. You know, there are different strain, Delta strain, Omicron strain, right? You must have heard of these. So there are certain viruses who mutate at a faster rate and they changes every time. So if they change every time, you will be needing the different antibodies. I told you, if there is an antigen, the antibodies are antigen specific. If it changes the antigen during every mutation, you need the new antibodies. Imagine the chickenpox virus changes in the second infection, these antibodies will not work. But this is not in the case of chickenpox virus, it does not mutate at a faster rate. But like other viruses, COVID virus, HIV, they all changes at a very faster rate. Right? Okay. So when people got to know, yeah, there is this kind of response called primary and secondary, they came up with the idea of vaccination. So what is vaccination? Okay, so vaccination or vaccine, the vaccine word came from vaka, and vaka means cow. Ma'am, what cow has to do here? Let me tell you what is vaccine first, so that you can understand why it has given the name vaka. Okay, so first of all, what is vaccine? Vaccine can be a dead or live or weak pathogen. So what we put in a vaccine? Basically the vaccine either contain live, dead or weak pathogen, or the part of pathogen, right? So whatever injection they are giving you, in that either it can be a part of pathogen or it can be the complete pathogen, but the pathogen either it will be dead. If it is live, that means if it is live, it's working, it's live, right? Then it will be weak. Okay, so it can be a part of pathogen or a dead part of the pathogen or dead pathogen. When it gets injected inside your body artificially get exposed with the antigen of the pathogen. Now the body thinks the pathogen is there, right? But it is weak enough to cause you a disease. The pathogen is weak enough to cause you a disease, but immune system thinks the pathogen has arrived. But we have deliberately made it dead or weak. We are just exposing the body with the antigen so that the body thinks the pathogen has arrived and it start producing antibodies and memory cell. So by giving vaccine, you are artificially generating primary immune response, so that for the second time when the actual pathogen comes inside your body, the immune system is already ready to kill it. So what we are doing, by giving vaccine, the dead pathogen, we are generating primary immune response, so that the secondary immune response will occur when you get actually exposed by the pathogen. Okay, so that's a fundamental. So it's basically nothing, it's the dead or live. If it is live, it will be weak. So live and weak pathogen or the part of the pathogen. It is injected inside our body to generate primary response, primary immune response. Okay. So why to take the disease and then generate primary immune response? Why not to take a vaccine? Just like that, some people get exposed with the chickenpox virus, they got immune for the second time, right? Now there can be another thing also. If one child has never been exposed to chickenpox, or they never, if there is a child of 5 or 6 or any years of age and have never got the chickenpox ever in their life, what we can do? We can give the chickenpox virus vaccine. Again, the same primary response will be generated. Why to give the disease? Give the vaccine and no disease, but the same primary response will be generated. Okay. Now, why it is known as vaka, or why we call it as cow, or why the word has been taken from cow? So that is in the discovery of the vaccine. The father of vaccination, is Edward Jenner. So Edward Jenner was the scientist who made the first vaccine, and that was against smallpox. So smallpox was a very problematic disease at that time, right? What happened in smallpox? Just like chickenpox, we had small pustules on our entire body, and but they are watery filled, okay? And whenever the person gets better with the smallpox after the disease, the pock marks are all over the body. And in some cases people also get blind if these vesicles they occur inside the eyes. So it's a problematic one. So what Edward Jenner did? Edward Jenner came with the idea. Edward Jenner was a keen observer. He saw whosoever people are living near cows, those who are cowboys and cowgirls, those people who are living near cows, they never get smallpox. Why? Because cow gets cowpox, and cowpox virus is a weak virus. When it entered inside the body of those cowboys and cowgirls, their body generate primary immune response. So when smallpox virus gets inside their body, the primary immune response is already generated. Yes, and now secondary immune response will occur, they will never get a disease. You didn't understand? Let me explain you. So imagine this is a cow. Okay, this is a cow. Alright. Now cow have cowpox virus in it. What is this? Cowpox virus. Now you must be thinking, ma'am, can cowpox virus generate the same primary immune response for smallpox? Yes, it can occur. Okay. Now there are people who are working near the cows. They also get exposed with the cowpox virus, but their body has generated primary immune response. Primary immune response. By the entry of the cowpox virus, the primary immune response is generated. Okay, imagine they get exposed with the same person get exposed with the smallpox virus. This is smallpox virus. So during exposure of the cowpox virus, some antibodies were generated. Now the same antibodies can also work for the smallpox virus, and this will be killed, and this will be completely killed, and this is how no disease. So what Edward Jenner did? Edward Jenner took the cowpox virus and injected into a 5-year-old baby, right? 5-year-old child, which is unethical right now, but at that time there was no rules and regulations, right? So Edward Jenner took the cowpox virus and for the experiment injected in the 5-year-old child, and saw that child got a little fever, but later on after a day the child completely recovered. Just like after every vaccination dose we also get fever, okay, because the immune system is working at that time. Now Edward Jenner exposed the same child by the same child to whom he has exposed with the cowpox virus, exposed the child with the smallpox virus, just like this. First he injected cowpox virus and then he injected with the smallpox, and luckily that child didn't get any disease. So by that it was stated that yes, vaccines work. So this was how the vaccine was originated, and smallpox vaccine was given to a lot of people, and nowadays smallpox disease is entirely eradicated. So that's why government is saying have vaccination, because if the complete people, if a complete herd it gets immunized, and that will be a better because the disease will not spread more, right? So this is how the vaccine took its birth. And now let's talk about what are the various types of vaccine. We have classified the vaccine into 3 generations. The first generation vaccine, the second generation vaccine, and the third generation vaccine, and the third generation. So just like your grandfather, your father and you, who is more modernized, your grandfather, your father or you? Definitely you are more advanced. Just like that, we also have different generation of vaccine on the basis of their advancement in their production. Okay, so firstly, the vaccine that was used was like a complete pathogen was used, as I have told in the discovery. So that's a first generation vaccine. In first generation vaccine, what we do, we basically use live and attenuated. Attenuated means weak pathogen. Either we use live and attenuated pathogen or dead or heat killed pathogen. Dead or heat killed pathogen. But the thing is, we use entire pathogen. We use entire pathogen to make the vaccine. So here entire pathogen is used. Entire pathogen is used. So you have to take the entire virus or entire bacteria. Either it will be live. If it is live, it should be weak. It should not cause you any disease. So that's why some vaccine, they are, should be made very carefully, because if it is live and you have not made it weak very nicely, they will cause you a disease. Okay, or it should be dead. Let me give you some example. OPV, oral polio vaccine. Oral polio vaccine contains live pathogen, also known as Sabin's vaccine. Whereas there is one polio vaccine which you inject, that's IPV, injectable polio vaccine. Here we use the dead heat killed virus, also known as Salk's vaccine. But ma'am, we have always heard that there are 2 drops of live, 2 boons in the gigi, right? We always have taken oral polio drops. We never have heard of this. In our country, the vaccine given is always oral, and in other countries it is given injectable. But if I compare both of them, oral is better. Why? Because polio virus, if you know, because I have taught you earlier, the polio virus enters through gut. If you take the vaccine through gut, it will also make your gut immunized. But if you take it through blood, your gut will not get immunized. So it's better than the injectable one. Okay, here the pathogen is live and here the pathogen is dead. In the second generation, we use only part of the pathogen. Here we are using the entire virus or entire bacteria, but here we are only using some part of pathogen, which is basically an antigen, because antigen will generate immune response. Why to take the entire virus if its only outer surface contains the antigen? Let's take that surface only, right? So only part of pathogen is used which act as an antigen. But the only condition is that you will use that part which is the antigen. Imagine the entire plasma membrane is the antigen, but you are taking nucleus. Then it will not be a successful vaccine, because antigen is the outer surface, that is the plasma membrane, right? Now, what can be the part? The part can be a toxin which is inactive. You call it as toxoid. So there are certain organisms, certain bacteria like cholera. The cholera toxin cause the disease. Okay, the cholera toxin cause the disease, not the entire bacteria cholera. So we will take the toxin of the cholera, make it weak, destroy it. You call it as toxoid and use it as a vaccine. So toxoid is basically inactive toxin. We can use it as a second generation. Okay, and the second is, it can be any cellular fragment. That means the part of a cell, like LPS, lipopolysaccharide layer, right? It can be anything like some proteins or glycoproteins. Anything can be antigen depending upon the particular type of what you have the disease. Okay, so here the example is like DPT. DPT is for diphtheria, pertussis and tetanus. This is one vaccine. Another example is hepatitis B vaccine. Hepatitis B vaccine. But hepatitis B vaccine is a recombinant vaccine. What is recombinant vaccine? I'll let you know very soon. In third generation, what we do, we use rDNA technology. We use vectors to make the vaccine. Example, hepatitis B. Now ma'am, now how both are there in the second and third generation? Let me explain you. We make hepatitis vaccine by 2 ways. One is a way which we are using nowadays, which is a second generation, but yes, that is recombinant vaccine, and we are using our DNA technology. Right? How? What we do, we take yeast. So it is always said this vaccine is produced in the yeast. And now we take the virus, which is the hepatitis B virus. And we know this portion of this virus is an antigen. And this portion of the virus, which is acting as an antigen, is produced by the genes. These are the genes. Now I took the gene out, put it in the yeast, gene of the hepatitis virus that produces antigen, not the any other gene, only that gene that produces the antigen. Okay. Now put it in the yeast will secrete antigen. Yeast will secrete antigen, and this antigen will be injected into the human as vaccine. I am not giving the entire hepatitis B virus. I am not giving the yeast to the patient or the person or the human. I am just giving the antigen to the human. And that antigen is the antigen of hepatitis B virus secreted by the yeast. Right? This is how it is second generation, because we are not taking the entire pathogen. If we are taking the entire pathogen, it will be first generation. We are just taking the part of the pathogen, so it is a second generation. Okay. What about the third generation? The third generation vaccine is still in the clinical trials. Third generation is still in the clinical trial. Clinical. Ma'am, but please explain us now how it's different. Okay, let me explain you. So what we do in this one is, we are taking gene from this virus and we put it in the weak virus, and this weak virus is a vaccine. Here we are just taking the antigen, maybe it's a glycoprotein only, and putting it inside in a body. But here we are taking the gene of the hepatitis B virus, put it in the virus, and then virus will be injected into our body. So this may be a dangerous one. Why? What if this virus is weak? You think is weak, later on undergo mutation and makes a virus that is deadly and cause you disease. That's why it is in clinical trials and not used nowadays. But this one, the second generation, which is produced in the yeast, secretes the antigen, is used nowadays, and yes, a lot of people take it. Okay, but it's also a recombinant vaccine because it is produced in yeast. I hope that's pretty clear to everyone, right? Okay, moving further to the next topic, and that is allergies. Ma'am, allergies, yes, we are very excited to know how allergies occur. Okay, sometime when you eat some food, packaged food, behind packaged food it is always written, if it contains some peanuts or so, it will always be mentioned it contains allergens like peanuts. So it's a kind of an advisory, or you can say a caution, right? That see, it contains peanuts, so you take care of yourself. Why? Because many people are allergic to peanuts. So what is allergy? Let's see. Allergy is also known as hypersensitivity. So there are some of your friends or relatives or so who easily get really very sick, and you say, oh, you are very sensitive, right? So allergy is that only. You are very sensitive to a particular product or a particular thing, and that particular product or a thing is known as allergen. My body to dust may not act weird, but to the same person, or sorry, to the another person, the same dust may generate an enhanced immune response. So you call it as exaggerated immune response. So what happen in allergies? We get exaggerated immune response to a particular allergen. What is an allergen? The substance that causes you allergy. Now the allergen can be a pollen, it can be dust, like in hay fever, people are allergic to pollen. So whenever there is a pollen season, people get lot of fever, running nose and also, right? Dust like asthma. People got asthma due to dust, but other person have no problem with that. Okay, there are certain food particles like nuts, cashews, or there can be certain drugs also, medicines, and like penicillin. Penicillin can cause anaphylactic shock. Anaphylactic shock is a serious serious thing of the allergy, the extreme case of allergy where people can die, and that's fatal. So what basically happened during allergies? Let's see. So imagine you get first exposure with the allergen. For the first time in life, I ate a drug. For example, a person got stomach infection. The person is very little, a baby, right? And we don't know what that baby is allergic to. For the first time, the baby is given any antibiotic or any drug, and the baby ate the drug. Okay. So what happened? Imagine the baby is allergic to the drug, but we don't know the baby took the drug for the very first time, right? So the mast cell, which cell? Mast cell gets sensitized. What does it mean? In the first exposure, your body gets sensitization. That means sensitization means it is getting to know that something is not good for you. Okay. I am just talking about people who are allergic. So what happened during first exposure? Antibodies are formed. But you will not get to know that you are allergic. Okay. So the antibodies will attach to your mast cells like this. You call this as sensitization. What do you call it as? Sensitization. Okay. Now what happened during second exposure? What happened during second exposure? Now baby took the same drug for the second time. Now you will got to know that the person is allergic. How? Now what will happen? These mast cells which are already sensitized, these mast cells which are already being sensitized, they will, or the mast cell which have been already been sensitized, they contain antibody, and the antigen or the allergen, for example that drug is an allergen, will bind to the antibodies like this. So here this is the allergen. Okay, in the first exposure allergen produce antibodies, in the second exposure allergen bind to the antibodies which are on mast cell. This will cause disintegration of mast cell, and mast cell will undergo degranulation. What is degranulation? Mast cell contains protein granules in them. These are small protein granules of mast cell. Now during degranulation, these granules will be exposed and they will secrete histamine. And histamine is the one that causes allergies and inflammation. Right? If it is very much enhanced, it can lead to anaphylactic shock and person can die. What happened? When you get extreme allergy, this windpipe, it got constricted and you will not able to get air properly and you will die. But as there is some problem, we always have solution. What's the solution? What drug can you give to a person? You can give antihistamines. Antihistamines will block the histamine and there will be no symptom of allergy. Right, like there are certain drugs, Allegra and cetirizine like that, right? Also we can give immunosuppressants like cortisol, immunosuppressant. It will suppress the immune response. As you know in allergy what's happening? Immune response is much higher. This is exaggerated immune response. Why not to suppress it by cortisol? We can also give the hormone like adrenaline, also known as epinephrine. Why? Epinephrine is known to do bronchodilation. It's a bronchodilator. I have told you your windpipe will get constricted, your bronchioles will get constricted, you will not be able to breathe. That's what happened in asthma a lot. The person is allergic to dust, dust go to bronchiole, bronchioles got constricted. And here if you give the epinephrine, bronchioles will dilate and a person will get saved. So all these things can be a solution. Any of these taken at proper right time can save a person's life. So you should definitely know what are you allergic to, right? Okay. Moving further to the next topic, and that is graft rejection and autoimmunity. autoimmunity. Graft rejection we have already done in the previous class. Whenever you got organ transplant, the graft or tissue of donor is rejected. Now who rejected? By T cells, and that's which response? Cell mediated immunity, right? So how can we suppress or how can we prevent the graft rejection during organ transplant? It's just a revision we have done in the previous class also. We can match or matching of MHC is required. If they matches, the rejection chances are very less. Second, we can also give cortisol. Why? Because cortisol is immunosuppressant. Because it will suppress the immune response and the rejection chances will also be less. So these two things are always taken in mind during organ transplant. Now what's autoimmunity? That we have also discussed in the previous class. It's a revision. I tell you it's a revision. Sometimes our immunity fails to recognize our own body cells and kills them by generating antibodies. For example, somehow if my body start to feel that the beta cells of my pancreas are foreign, the body starts to feel like, the immune system starts to feel like that beta cells of my pancreas are foreign. Now immune response will be generated against it and the cells will be killed. I had given you homework to tell about all the autoimmune disease. Let's discuss them today. Now let's make a list of various autoimmune disorders. Okay, let's get started and start learning with me. The first one is type 1 diabetes. In type 1 diabetes, your beta cells are destroyed. So here I am writing cells or organ which got damaged due to your autoimmunity. Okay. So which cells are destroyed? Beta cells. Of which organ? Of pancreas. So your body starts recognizing your beta cells of pancreas as foreign, right? Second disease is your psoriasis, which we have discussed yesterday also. Here the skin cells are destroyed. Then we have vitiligo. Here also the skin cells, but which cells? Melanocyte, the melanin secreting cells. Melanin is a pigment. They are destroyed, and that's why somewhere skin is pigmented, somewhere it is not. Then the fourth one we have is rheumatoid arthritis. In rheumatoid arthritis, your joints are affected. What are affected? The joints are affected. Then we have another disorder which is multiple sclerosis. Multiple sclerosis, where the nerve cells are destroyed, and this is dangerous. If nerve cells are destroyed, then it's very bad. What in nerve cells are destroyed? Myelin sheath. Then we have lupus. In lupus, multiple organ can be damaged, one or multiple, or any organ can be damaged. For example, the kidney is damaged, just like that, or a liver is damaged. So these are some of the autoimmune disorders, and you have to learn each and every disorder, because sometimes they will simply ask you a question, find the autoimmune disorder from the following. You can easily find that out, right? We also have some other disorder for thyroid, like Graves' disease and thyroiditis. Both are for thyroid. But the difference is, here the thyroxine level, thyroxine is a hormone, increases, but in this one thyroxine hormone decreases, right? And the reason is because here the cells which are secreting thyroxine is destroyed, but here the receptors for the TSH hormone is destroyed. So these are the various autoimmune disorder. Let's talk about lymphoid organ. So we have a lymphatic system inside the body. You must have heard of a lymphatic system and vessels in the chapter body fluids and circulation. So there are lymphoid organs associated with them. Just like for circulatory system we have heart and blood vessel, for respiratory system we have lungs, for your digestive system we have gut, for your immune system we have lymphoid organs. So these are the organs of your immune system and they are of two kinds. One are the primary lymphoid organ and another are the secondary lymphoid organ. What's the difference? Let me show you. For example, the primary lymphoid organs are organs where your main immune cells, the B cell and T cells, they are formed and get mature. But the secondary lymphoid organs are the organs where those mature cells will go, they will identify the pathogen and they will kill it. Just like your college, your medical college is a college where you learn everything and you become a doctor. But where do you work? You work in a hospital. So hospital is a secondary lymphoid organ. Fine. So in primary lymphoid organ, it's a site of formation and maturation of B and T cell, and they are the site of action where they will function, right. So there are two cells that are very much important. One are the B cell, another are the T cell. If I talk about the formation of these both the cell, okay, I am writing here. If I am talking about the formation of these two cells, both the cells, they are formed in bone marrow. B cell is formed in bone marrow and the T cell is also formed in bone marrow. Okay. If I talk about the maturation of these cells, the T cell, they got mature in the organ named as thymus. So thymus and bone marrow both are your primary lymphoid organ, whereas these one, they get mature in bone marrow. But the B cell, they divides in thymus because they have to undergo division on exposure to the pathogen. Only then B cell will get turned into plasma cell and memory cell. So their division occurs in the thymus, but their maturation and formation both occur in the bone marrow, whereas the maturation of T cell occur in thymus. That's why thymus, the T cells, they got a T from thymus, and B cell got its B from, not from bone marrow, from bursa of fabricus. Bursa of fabricus is the bone marrow of bird or aves. It's a bone marrow of the aves. Fine. So this is how it got its name, B cell. Now what are the various secondary lymphoid organ? We have various type of secondary lymphoid organ. We have spleen, we have lymph node, we have liver, we have appendix, we have malt, we have Pierce patches. All these are your lymphoid organs. Alright, let me take it one by one. Spleen. Spleen is the organ present behind the stomach. We will discuss that in detail. Lymph node are small nodule-like structure. They are present here like these. In the groin region, that means in between the legs, and move downwards towards thighs. And also they are present around armpits and the neck region. So all these are lymph nodes, as you can see. Okay. Then we have liver and an appendix. Yes, they also act as a secondary lymphoid organ because they have macrophages in them. Malt is mucosal associated lymphoid tissue. If you remember, malt is present in the mucosa of your GIT, gut. Yes, and wherever you will get mucosal lining, whether it is respiratory tract, urinogenital tract or digestive tract, you will get malt. So we say around 50% of all the lymphoid organs in your body is your malt, right? Pierce patches, they are present in your ileum, right? Ileum is a part of your gut. Ileum is a part of your gut, right? So let's talk about spleen that you don't know about. So spleen is present behind stomach. If this is your stomach, imagine this is your stomach. So this is present behind the stomach. This is your spleen. So spleen is present behind your stomach. Okay. Now the spleen has two areas. It has two areas. If you will cut the spleen, you will see two areas. One is white in color, another is red in color. The white one is responsible for all the immune response. It contains B cell and T cell, right? Whereas red area, it contains RBC. Here the excessive RBCs are stored and it's also the graveyard of RBC. That means RBCs are destroyed here. So do remember, yes, spleen is a graveyard of RBC, but the RBCs are also stored here. And white region is all for immune response. How it works? Imagine this is a spleen, it's a nodular structure. Just like any other organ, it will also receive the blood supply. So artery will be bringing the oxygenated blood and vein will be carrying away the deoxygenated blood. So with blood comes a lot of pathogens, impurities and pathogen. Imagine these are the pathogens that are coming. Now the spleen have the immune cells. Immune cells will take away the pathogen, and whatever blood is moving, it does not contain any pathogen. So we say it's acting as a filtering organ. Just like you filter tea, right? You put the tea, all the tea comes in the sieve and clear tea is in the glass, just like that. So we say spleen is a kind of a filtering organ. Just like this lymph node works. Lymph node also act as an filtering organ. Okay, so that's about your lymphoid organs, and let's solve some PYQs. Important. First, which one of the following statement is correct with respect to immunity? Okay, you have to tell which is absolutely correct. Preformed antibodies need to be injected to treat the bite by a viper snake. Okay, you have to find the correct one with respect to immunity. Antibodies against smallpox pathogen are produced by T lymphocyte. Antibodies are protein molecule, each of which has 4 light chain. Rejection of kidney graft is a function of B lymphocyte. First is absolutely correct. If you get the injection of, or you get bitten by any poisonous snake, venom will be there. We need some antibodies to kill it. Antibodies against smallpox are not secreted by B cell, they are secreted by T cell. Smallpox is just an example they have given. Antibody is a protein molecule which contains 4 chain, which are 2 light chain and 2 heavy chain. Rejection is by T cell, not B cell. So answer to this question is 1. Next, malt constitutes about percentage of the lymphoid tissue in human body. Is just now we have done this number 50%. So malt is 50%. Next, identify the wrong statement with reference to immunity. When ready-made antibodies are directly given, it is called passive immunity. You have to find the wrong one. Okay. Active immunity is quick and give full response. Fetus receives some antibody from mother. It is an example for passive immunity. When exposed to antigen, antibodies are produced in the host body. It is called active immunity. So in the first one, if you will see, the ready-made antibodies are given and it is passive. The active immunity is never quick. It takes time. Right, it's never quick, it takes time. Primary response takes time. So the statement is incorrect. Answer is 2. Fetus, yes, it receives antibody, it's passive immunity, and whenever you're exposed to antigen, you produce antibody, that's the active immunity. So wrong is second statement. Next, antivenom injection contains preformed antibodies, while polio drops that are administered in the body contain. What does it contain? The polio oral one, it contains live but weak pathogen. So it is the attenuated pathogen. Attenuated is another name for weak. Answer is 4. Alright, next question. In higher vertebrates, the immune system can distinguish cell, self and non-self. Okay. So it is saying that our cells know which are their cell, which are not their cells. If this property is lost, so now body cell doesn't know which is our own body cell. Due to genetic abnormality and attack self cell, this leads to autoimmune diseases or autoimmunity. Answer is 2. In allergy, though allergy and autoimmunity both are genetic. Yes, both are genetic. So that's why they have written genetic. Both are genetic. But in allergy, it's the hyped response. But here, in autoimmunity, your own body cells are attacked by your own immunity. So answer is 2. In graft rejection, it's organ transplant. In active immunity, it's quite different. So answer is 2. Which of the following immunoglobulin constitutes the largest percentage in human milk? Very simple, IgA. Next. That's it. So that's it about the immunity part at all. So in the next class we'll be discussing about the diseases. It will be like AIDS or cancer, any of these, so it's a surprise. So I'll meet you in the next class. Till then, keep on studying and please do read the NCERT because immunity is very, very, very important.